is a principle. Okay, principle of Mendelian genetics. Nanti kita tengok what is the principles behind it. But this is the person behind it. Okay, Gregor Mendel. Nama pun lagi Mendelian. So the Mendelian genetics is based on his name, Gregor Mendel, an Austrian monk, and he spent 8 years doing experiment with peas. I think this story pun you all dah already being exposed to, I think in the SPM ataupun during your bio class during matric kan. Siapa Gregor Mendel ni, what has he done with the experiment. And if you want to know further, you can just Google lah apa Gregor Mendel ni dah buat. Okay, how did he do the cross breeding between the peas tu, so that he can get a lot of variation in the phenotype. Okay, so what is phenotype? Nanti kita tengok what is phenotype, what is genotype. Okay, and he proposed these principles of hereditary in 1865. Okay, so he studied 7 characteristics in peas. So if you look here, these are the characteristics, or we can call it as phenotype. Okay, phenotype ni contoh yellow seed. And also some of the peas are having green seeds. Some of them have round seeds. Some of them have wrinkles. Okay, so these are actually the phenotype from that characteristics of that apa tu animal ke ataupun this plant. Okay, some of them have a yellow pot, some of them have green pot. So he did a lot of crossbreeding between he how that happen, why there are variations in this. Okay, and if we look at humans and the variation our same ethnicities, even we cannot find the people who have the same character unless they are twin lah kan, unless they are twin yang sharing the same if they are twin but they are from the different placenta, then they come out differently juga. Okay, so only twins that have the same genetic component, genetic features or genetic how do you call it, genetic profile. Okay, so he did a lot of this apa dia tu, he did the crossbreeding tadi kan, and then he come up with this Punnett square. I think you have heard Punnett square before. So Punnett square ni is the way that we can actually determine how many percent that the offspring is going to have the certain characteristic. So contoh sini, dia kacukkan the S besar is call it it's representing allele. So each of the cell macam kita pun sendiri, we have 2 allele because we have double strand of the chromosome. So each have the same have their own allele. So kita ada 2 set, satu set of allele. 2 maksudnya 2 allele lah, 1 set. Okay. So for example, in these 2 yang dia nak kacuk ni, we call it macam parental generation ni, the one it has capital S, the other one has small s. So this allele and then this allele, and then the gamete only take one of it. So ada satu lah allele from each mother and father tu. So ada satu capital S and one and the offspring of F1 generation, it's capital S dengan small s kan. So we call it as heterozygous. because it has two different allele, heterozygous. If for example, the father tadi tu capital, we call it as homozygous. Okay, the mother pun small letter, we call it homozygous because it's similar. So the offspring here has the heterozygous allele. Katakan dengan the offspring also have two generation. Some of it have capital, homozygous, some of it heterozygous, and some of it homozygous, small letter. So he come up with this Punnett square based on his observation. Dia tengoklah berapa banyak yang dikasutkan tu, berapa banyak offspring yang ada this characteristic. So it makes it easy to determine how many percent that can ada certain. So we can do this together. For example, allele tadi, the capital B. So capital B represents straight hair, contoh. Okay, and then small letter b represent curly hair. We look at our father and mother, for example. So if the mother has homozygous for straight hair, capital B, capital E, so we call it as genotype lah. So actually this one is the genotype. The apa allele yang ada tu, combination of two allele tu, we call it as genotype. So the mother has a genotype of a straight hair, homozygous, and the father has heterozygous genotype, straight hair and curly hair. Okay, so the father, capital B and small letter b. So sekarang we want to know if they have a child, is the probability of the child having wavy hair. Yang ni bukan wavy. So 50% of how many probability of a child having a curly hair? What is probability of having a child with is the sentence? This one. Okay, so it's B. So one from here. This will be E. So this one will be capital. Probability percent having a child with curly hair. Anyone wants to try? You can unmute or you can put in the chat. Waimun 50%. Shaza 50%. Iwais 50%. Shanyi 0%. Ada yang 0, ada yang 50. Macam mana tu? Macam mana tu? So, provided that, yang ni one information that I did not disclose, provided that the recessive allele, the B is the recessive allele. Recessive meaning that recessive tu maksudnya they need to be present in two allele in order for it to be expressed. So that is recessive. So imagine this is B, small letter B is recessive. So it needs to be kita punya ni express homozygous in order it's present as a phenotype. So if I make like that, kita tak BB, small b, and so it's actually 0%. Okay, because of the allele tu is capital. Whenever it is present, all so the child will have that phenotype of straight hair. Okay. So yang ni 0% lah. But what is the ability of having a child with straight hair? 100%. So satu lagi situation in which if mother has B and also small b, the probability kan berubah B besar. This one would be capital small. Okay, so in this way the answer would be different. Okay, so now what would be the answer for the curly hair? What is the probability? %. All right, so 25%. Oh, probability tu macam 25% lah. So kalau anak dia lahir, there is 25 times chance. Just this one lah. Okay, BB and also BB, semua BB kan. So capital and also small letter. So these are the homozygous. Heterozygous would be the combination lah. And also. Okay. All right. Semua okay? So Mendel's law, we have three. Mendel's law of segregation. For segregation the from each gene. So itu betul lah. Segregation sebabnya sekali berlaku production and then only one is will be transferred to the gamete. Okay, so of segregation. One law of independent assortment. Genes for different traits can segregate independently during the formation. Therefore, inheritance of genes at one location does not influence inheritance of genes at the location. Sebab kita ni kan panjang. So we have we will look later on in the activity berapa panjang kita punya genes ni. And then so the segregation in one location is independent to the segregation at another location. So diorang macam tak it does not have macam it has to follow certain location tu semua kena segregate at one order ke apa, tak ada. So it's independent. And then the third law of dominance, some are active, and organism with at least one dominant allele will display the effect of the dominant allele. So it's like tadilah recessive ataupun dominance. Kejap. Oh, okay. Alright. So maksudnya law of dominance tu maksudnya the one phenotype that is present um, is the dominant one from that allele. Contohnya tadi kan we have heterozygous, tapi which one actually? Is it curly ke is it tu? Kalau yalah of the man, there is an exception in which does not follow the Mendelian law, in which contohnya there is a situation in which contohnya the flower, the color of the flower is white and another allele brings the color of red, for example. But then tiba-tiba kita kacukkan yang putih dengan yang merah, and then the offspring color. So this is what we call incomplete dominance. Maksudnya dia akan jadi something else, different color. Okay, so this is actually deviates from the Mendelian law. Tapi it Mendelian law yang mana yang biasanya lah assumption. Okay. The second part, which is the structure and components of the gene, okay, DNA and the gene. So if you look here, this is the nucleus. I think you all pun dah tahu semua ni kan, belajar banyak kali kan. So in the cell, we have macam-macam benda lah. Mitochondria, apalah semua. And we have the nucleus, okay. So in this nucleus, our DNA profile. Our genetic profile is stored in this nucleus. Okay, every cell in our nucleus and genetic profile. So in the chromosome helix, double helix is inilah the structure. We call it double helix. Dan dia berpintal-pintal. And in the end, it form this chromosome yang bentuk macam X ni. Okay, so if you open satu-satu of this strand, dia macam tu kan wire yang telefon rumah tu. Okay, and then from this, actually the way that it curl, actually it makes it more macam compact. Okay, so that's why it has this kind of feature, and it was being organized. I mean, this is the histone. It's macam setiap a few a few jarak tu they have the histone. It's like a compilan of this genetic strand, and you open it again, open the double helix, and you can find the. Now we come to the activity dulu lah. Okay, so now is more towards the recap of what you have studied before. So if you open the Mentimeter, I will also need to try it. Dah lama tak guna. If you can try it with your phone and see how it goes, whether you can open this. And let me know if you have problem, yeah? So I will need to. Open this. Yeah. Okay, now we have 63. I think we're waiting for more. Okay, alamak I dah tekan. So you play je lah ya. Okay, so I think sekejap ya. I will share another screen. Okay, correct, correct. Ada yang belum masuk ke? Let me check whether I can. Sekejap ya. Ada yang tak dapat masuk, let me know so that I can give you the link before I proceed. Have course je kan? Okay, alright, good. Okay, so continue. Okay, of genetics. Who is the father of genetics? Jangan tengok jawapan dia. I know I share this. Tapi try to um, answer on your own dulu. Okay, Gregor Mendel, yes, he is the father of genetics. So Francis Crick and James Watson, nanti kita tengok what are their contribution. But if you say father of genetics, then it would be Gregor Mendel. Okay, next question. Sekejap. What are genes? Okay, this one we have a time. Genes are a code of DNA. Okay, so we can call it as a code of DNA lah. Okay, double helix too, it's the, how does it look? So it is in a double helix. So it's like a recipe book, human, any other. Okay, next question. What are DNA made of? So it's made of nucleotides. Okay, 70 of you. Betul. Amino acid, ni 7 orang ni. Amino acid tu, if you change the question to be, so what are proteins made of? Then you are correct. Proteins are made of amino acid. Tapi DNA is before amino acid is being, apa tu? Okay, is before that. Okay, your nucleotide and your DNA are your recipe. So dia punya amino acid tu dah jadi kuah dah. dah jadi makanan dah. Okay. Okay, enter. Long pieces of DNA are organized into individual units called okay. Long pieces of DNA are organized into individual units called chromosome. If you remember the picture just now, yang bergumpal-gumpal, nanti in the end dia akan jadi chromosome berbentuk X tu kan. So, all of the long piece of DNA actually di dalam that chromosome. So, cell tu dah already a bigger unit dah of that. Okay. Okay, next. So, DNA is copied to make an mRNA. So, what is this process? Okay, DNA is copied to make an actually DNA transcription. Nanti kita akan revise balik, okay? So yang ni 14 orang ni memang dah tak ingat dah lah apa yang dia belajar dulu ya. Okay, DNA translation is something else. Okay. mRNA is decoded into amino acid. So what is this process called? I share ni ke? I share. Oh, I share screen lain rupanya. Kejap ya. Okay, so this is the screen that I supposed to share. Sorry ya. All right. So the mRNA is decoded into amino acid. So, alright, DNA. Tadi kan dah jawab dah DNA transcription is daripada DNA transcribed into mRNA. So now mRNA is decoded into amino acid. This is DNA translation. Okay, we translate from the genetic code into amino acid. So that's translation process, okay? RNA splicing is something else. Okay, removal of introns and joining of exon in the mRNA. So what is it? Which process involves this? Okay, removal of introns and joining of exon in the mRNA. So yes, correct. It is RNA splicing. Okay, nanti kita tengok further. Okay, so dah habis dah soalan. Okay, tengok leaderboard. Siapa nombor 1? Po. Yeay, siapa Po? Kat tangan. Walaupun saya tak nampak. Congratulations Po. And then Hui Ai, Eric, Ashley, Yijun, Azman. Okay, so very good lah yang mana yang boleh jawab. Kalau tak boleh jawab tak apa. Kena study lagi. Okay. So this is just a basic recap sahaja. Okay, on previous knowledge. So now we have a look at the have a look at each of these component of DNA regulation. Am I sharing now? Not yet. Okay. Okay, nah, kita continue with this. Okay, yang ni, this is only the definition. Okay, your definition of the terms. And this one you should know already by now. It's just a recap. Chromosome tu apa? Okay, you remember the X. X punya bentuk tu, the chromosome. Gene is actually the term in which we actually want to associate that with the phenotype ataupun apa gene tu, because within our chromosome it actually holds a lot of genes. If you say one gene, sebenarnya one gene is encoding for one characteristic or one phenotype ataupun one protein ke apa contohnya. Okay, so contohnya my gene, I have a gene of black eye. Okay, so that one gene actually contribute to the black eye, warna mata, okay, the color of the eyes. Okay, I have another gene that contribute to the apa tu, hidung mancung contohnya. So that's another gene. So all of the gene that characterize us as a human are all within that chromosome. Okay, and then DNA itself is deoxyribonucleic acid. So the DNA is a term in which we use, okay, as a general, DNA itulah kita punya recipe of life. Okay, the recipe of us, of all the other of the living things. Okay, all have DNA. So nucleotide is the building block for DNA. It has phosphate backbone, sugar, either one of the nucleotide bases, ATCG. Okay, nanti kita tengok the ni, ataupun you can recap back from the previous slides. We have the structure of the DNA tu sendiri. Okay, so now we come to the history sikit, the genomic DNA. So who are the contributors of this structure of the DNA? Siapa yang menemui? Okay, we have Frederick Meister extracted DNA from the nuclei of human white blood cell. So he extract, okay, and then the structure, waktu tu they don't know the structure, but he extracted that DNA from the nucleus. And then later on, Rosalind Franklin, she actually found the photograph of the image of the DNA. So she used, I think, the X-ray crystallography to elucidate how does the DNA looks like. So ini dia punya photograph lah of that X-ray diffraction image of DNA. So dia nampak, oh okay, berbentuk X macam tu. Okay, and then so she found out that it is a helical structure. So dia boleh nampak sahajalah. So until that, James Watson and Francis Crick and Maurice Wilkin has confirmed that, okay, this is the structure of the DNA. It is double helix. And they have published these findings and they get the Nobel Prize in physiology or medicine in 1962. But Rosalind Franklin is actually the pioneer of figuring out that DNA is a helical structure. Okay, but then she died before the rest of the team got the Nobel Prize. So she did not get the Nobel Prize. There is actually a drama in the history. So if you want to know further what drama it is behind this, so you can just Google lah, tengok baca sikit story dia. So this is the structure of the DNA. We have ATCG tadi kan, the nucleotide ATCG. So the whole strand too, either side of it, we have the sugar phosphate backbone. Ini kan, the blue one. And then these two backbone was interconnected by this combination of nucleotide base. So A will always be paired with T. You see here, thymine, adenine will always be paired with thymine. And C, cytosine, will always be paired with G, guanine. Okay, ATCG. So inilah the phosphate base, and then ada sugar in between phosphate dengan nucleotide base. Okay, so itu ialah basic structure. Jadi sama je tadi. If we pisahkan, if we take part, this is the phosphate backbone and this is the nucleotide base. And then lagi separuh tu ialah the complementary bases. Okay, A will always be paired with T, C will always be paired with G. Okay, gene expression, transcription and translation macam tadi lah. Okay, transcription, DNA sequence is copied to make an mRNA molecule, and then translation is actually when mRNA tu is being decoded into amino acid. So you have to understand the flow. Nanti kita tengok later. Okay, first, which animal has the same number of chromosomes as human? Boleh teka tak? So this is butterfly, chimpanzee, cat, or zebrafish. Ini tak payah Google lah, just teka je. Okay, which one? Have the same number of chromosome. Tadi number chromosome kita berapa? 46 kan? Okay, which animal has the same number of chromosome? Jangan Google ya, tak cik. Chimpanzee, Farzana kata chimpanzee. Hamisah pun chimpanzee, okay. Mana human kita? Oh, ada human kita. Okay. Siapa pula? Which animal has the same number of chromosome as human? Semua jawab chimpanzee. Actually, it is... Okay, siapa? Satu lagi jawapan. Before I reveal. Number ya, bukannya resemblance ataupun similarity of the component. Satu lagi jawapan. Semua sama. Lagi 10 minit lagi. Tak ada, semua chimpanzee. Tak ada ya. Okay, it's actually zebrafish. Okay, zebrafish has the same number of chromosome as human. 46 chromosome. So that's why zebrafish ni was being used in research juga. Okay, so there are some characteristic dia yang menyebabkan dia boleh digunakan sebagai research lah. Animal for research. So chimpanzee actually has I tengok nota lu 48 chromosome, but it has 98.8% similarity with human. Tapi number of chromosome dia 48, kita 46, okay? Cat ada 38 chromosome, butterfly has a variation of 5 to 300 chromosome, number of chromosome, depending on the jenis, type, okay? Alright. So at least kalau you tak dapat apa-apa pun from this lecture, at least you know lah, okay, you have the similar number of chromosome with zebrafish. And chimpanzee has 98.8% similarity with human chromosome. Tapi janganlah tu je yang awak dapat from this lecture, okay? So hopefully you get the other part as well. Okay, so how is gene regulated? Okay, transcription, DNA to mRNA, and then splicing. Okay, we'll see later what is splicing. And then translation from mRNA tadi to amino acid, which is later it will form protein. And then later on, it does not end there. That protein too, it needs to undergo post-translational modification before it can give function. I had a YouTube video you can watch later because we don't have much time. So I also put in the e-learn that video. Professor Dave, kalau tak silap nama dia. So he recap a bit in about 6 minutes about gene translation and transcription. Sorry, transcription and translation. Okay. Okay, baca jap this one. Okay, what does it mean here in this cartoon? Why is it important to have for the gene to be self-regulated? Self-regulated maksudnya gene tu kita tak payah nak bagi tahu gene. Kalau buat balik ni translate lah lagi, buat transcription lagi, kita tak payah bagi tahu because it's regulated sendiri. Okay, why is it important? Because here, if you can see here, is they are making the hydrochloric acid, but then they actually location dia salah. Okay, kalau dia tak regulated, maksudnya dia akan jadi things like this lah. Dia location dia dekat eye. So they produce, instead of dekat stomach, they produce dekat eye. Okay, that protein or that, ialah that protein diproduce kat tempat yang salah. So that makes it very unpleasant. Okay, it disrupt with the whole human function. So this is an overview. Okay, sama juga exon and intron. So each DNA, each gene ataupun the whole strand of DNA, we have different genes alongside. So each gene will have intron and exon region. Exon region is the one that are having that that are coding lah, yang boleh buat coding of that amino acid. But intron region actually tak ada that coding. So it's actually macam filler, okay? So during the transcription, exon and intron will both be transcribed. The exon will be transcribed. And then during the translation, there will be RNA splicing. So if you read here, some regions during the transcription, some regions are removed, which are the introns, and then the remaining exon are then spliced together, and the spliced mRNA molecule is prepared for export. So splicing tu maksudnya dia removekan all the yang non-coding region tu, and then it will be compiled only the mRNA yang coding tu saja, okay? And then it will be exported to the nucleus so that it will undergo translation, and then translated, the translation to the outcome there will be from each of the codon, it will be translated into different amino acid accordingly, and then this amino acid will be joined together to form the protein. Okay, so that's the basic of it lah. Okay, generally. So RNA splicing again is a process that removes the intervening non-coding sequences of genes, which is introns, from the pre-mRNA and joins the protein coding sequences exons together in order to enable translation of mRNA into protein. Okay. So ini contohnya pre-mRNA tadi, because DNA transcribed into mRNA together with the intron section, exon dengan intron together, but then the mRNA tu, before the translation, dia nak translate, of course dia nak translate encoding region sahaja, so they will remove, the intron will be spliced out, so the left, they'll be only left with the exon region in that mRNA strand, okay? Barulah dia boleh keluar ke nucleus to undergo translation. Okay, exon, intron, itu the differences, and the translation process. I think this one you can recap back, do your own reading, the translation process, macam mana, how does it start, what are the enzymes involved in this translation and transcription. And then you have to understand lah, the tRNA, what is the function of tRNA, okay? Which is the start codon, which is the stop codon. For example, here, the start codon here is the AUG, for example. If the tRNA reads that, the small unit of this of this ribosome reads this AUG, then it will acknowledge that, okay, I'm going to start the translation because there is a start codon. So it will continue forward according to the sequence of that mRNA. Okay, so it will create that amino acid in which the amino acid will then be forming the proteins. But before it is useful or being used for cellular function, it needs to undergo post-translational modification. For example, glycosylation, acetylation, alkylation, and methylation. Maksudnya you kena train dulu lah. So you imagine this is like a training untuk that protein to do their function. So kena gilap sikit, kena train sikit so that it will be able to do their function in the body. Okay, contohnya proteins ke, lipids ke apa. Okay, whatever transporter proteins. So it is actually after they have undergone this post-translational modification. Okay. Alright, ini tak ada apa dah. Alright, I think that's all to recap. I put some reading materials in the link in the slides. So I will give the slides later. I will post in the eLearn. And also the YouTube video in which you can have a look and you can use that as your references as well. Okay, so for you to recap on this topic. All right, any question before we end? Okay, for the exercise, you can answer the questions. I think the same questions that I post in the eLearn. And then if you want the answer, I think it's very simple lah. If you can just get your answers on your own. But if you want me to give an answer, just let me know. I will post in the eLearn as well for the answers. Okay, so I will see you in the next lecture next week. Okay, so next week we'll have 28. We'll have a face-to-face lecture in the DK. All right, thank you very much. Tak ada soalan lah. All right, thank you very much. See you next week. Okay, bye-bye. Assalamualaikum.
Thank you, Doctor.
Welcome.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doctor.